Go live. Very good afternoon, everyone. Now I request Dr. Grace Ratnam Madam to introduce the speaker, Professor Dr. Krishnan Sir from College of Pharmacy, SRIPMS. Grace Ratnam Madam, you can introduce Sir.
Good afternoon to one and all present here this afternoon for a series of webinar series conducted by Dr. M.G.R. Medical University. The topic which is chosen for today is a very state-of-the-art topic, converging towards improved healthcare. Living in a technological age, every day we are coming out with new technologies appear. So the whole idea of today's topic is how to converge different technologies from different disciplines so that finally we have improved healthcare. So this will start from digitalization, genomics, proteomics, gene therapy, and we also have personalized therapy, and we have seen different routes for vaccine therapy, computer-aided drug design, and nanotechnology. Today's speaker will also be talking more on nanotechnology, which opens new avenues for research for biologists. And so, going on to our speaker for this webinar, we have Dr. S. Krishnan with us. Dr. S. Krishnan is presently Professor and Head, Department of Pharmaceutical Technology at Pharmaceutical Biotechnology at College of Pharmacy, Sri Ramakrishna Institute of Paramedical Sciences, Coimbatore. He has excelled throughout in academics. He completed D.Pharm from Annamalai University, securing the 3rd rank, and then he did his B.Pharm from Annamalai University, securing the 3rd rank. M.Pharm, Pharmaceutics from College of Pharmaceutical Sciences, Manipal. There again, he secured the 1st rank at Manipal University. And later he completed PhD at 2009 under Dr. M.G.R. Medical University. He has over 33 years of teaching and research experience, having guided over 35 M.Pharm students and having more than 50 publications. He was also awarded Best Paper Award at various national level conferences. He is a PhD guide with Dr. M.G.R. Medical University and various other universities. He has been appointed by the National Testing Agency as a question paper setter and also by the selection committee by the DME of Tamil Nadu. He has been a resource person, delivered many invited lectures at AICTE sponsored staff development programs. He has participated in national and international conferences in capacities as chairperson, co-chairman, and member of the Scientific Services Committee. Award for 2009 by Manipal College of Pharmaceutical Sciences, Manipal. With this, I hand over to Dr. S. Krishnan. So he will be talking on the topic chosen for today. And after that, the session will be open for any questions. Over to you, Dr. Krishnan.
Thank you, Dr. Grace, Madam, for the elaborate introduction. And good afternoon to Honorable Vice Chancellor of the Tamil Nadu Dr. M.G.R. Medical University, Registrar, Tamil Nadu Dr. M.G.R. Medical University, all the other officials, principals of my college, Vice Principal of my college, and moderators, Dr. Grace Ratnam, Madam, and Dr. Sampath.
Natarajan, CECP.8.
Hello, is the audio clear, Madam?
So 1 minute, let me announce. Others can mute your audio, please, because people are listening, so you can keep yourself muted. So whenever you have questions, you can unmute and then ask the questions. Thank you.
Thank you. And extending with the welcome for all the principals and the faculty of the Tamil Nadu Dr. M.G.R. Medical University. Right. Very good afternoon to one and all here. And the topic which I have taken for this series of webinars being conducted by the Tamil Nadu Dr. M.G.R. Medical University is on converging technologies towards improved healthcare. Now, this word is self-explanatory. The sentence itself is self-explanatory. With the convergence being the order of the day, we need to converge on all the latest technologies with an aim of improving the overall healthcare. Now, with this, I move to the next slide. Yeah. So we'll start with a few of the quotable quotes. Being in the teaching profession, I thought we would correlate ourselves with education. So the aim of education is the knowledge of values and not of facts. So it has to be a value-based education. And the science of which we are all graduates and basic sciences students is organized knowledge, and wisdom is organized life. So usually a successful person is said to be a wise person, right? That's what this quotation really explains. And education is learning what we didn't even know that we didn't know. So this is removing the ignorance from the minds of students and the others. So this is the job of a teacher. And if you have the knowledge, let others light their candles to it. So with this quotable quote, we'll move on to the topic and we'll see what exactly is the meaning of this convergence. And the basic disciplines which have been converging for several decades now is the two important aspects, that is science and technology. If you see the slide on the left side of the slide, you'll see that science, what does science refer to? Anything which is naturally present can be termed as science. So there is some scientific principle or basic principle involved in that particular happening. It could be a functioning of a cell, which is the basic structural and functional unit of a life, right? So it could be a cell, or it could be a law of gravity, or it could be a law of buoyancy, anything which is has science attached to it is termed as science, right? Scientific basis. And what is technology exactly? The figure which is given on the right is the launching of a spacecraft. Technology is learning how something works and putting this sound theoretical principles into practice on a large scale. So you have to use this technology in a large scale and then integrate both science and technology to benefit mankind. How does this really benefit mankind? Does it all the integration benefit mankind? Yes, this is a classical example. Science and technology results in improvement of nature and great service to the mankind. Again, here you see the figure on the left side. You have a space shuttle, remote sensing space shuttle, which is able to give idea about the weather and whatnot, right? You have a space shuttle which can give you so much of information that is related to the technological aspect. And to the right, I don't know how many of the students are able to identify that. It is the three-dimensional image of the maxillary part of the body. So it's a 3D printing, which is now the buzzword, which is now available in market, right? So this is called as a 3D printed maxillary section. So this is how science and technology is able to develop the overall areas. Right now, this is the most important part of the talk, the scheme. One slide refers to all the topics which will be given an overview during the talk, right? Starting with the basic sciences, namely the physical sciences, chemical sciences, and the biological sciences. And then you have the analytical sciences and the computer sciences, material sciences. And the ones which are on the right are all little advanced fields like nanotechnology, system biology, microfluidics, bioinformatics, and various omics, right? These are all the advanced fields. And to the left, you have very important disciplines, namely science, technology, engineering, and maths. Together, they are called as STEM concept. So this is very prevalent in most of the developed countries, which is being emphasized now in today's India's system of education. Right? So STEM refers to science, technology, engineering, and mathematics. And this has now been upgraded to a STEAM concept, where the A refers to the art. So the art includes communication as well as scientific writing. So you need to communicate and to have the knowledge of learn the art of communicating to become effective STEAM practices. So the concept of STEAM has to be excellently inculcated in all the young minds, right? So this is with regard to the conventional methods. And the one which is seen at the bottom of the slide, these are all futuristic concepts. One is the digital and virtual healthcare, about which during the COVID times we have seen a lot about how digitalization and virtual healthcare has helped in overcoming the severities of pandemic COVID-19. And then you have this important term, which is NBIC, which combines the technologies of nanotechnology, biotechnology, information technology, and cognitive sciences. Now, nanotechnology has been the buzzword for the last few decades. Biotechnology, again, last maybe 4 decades, we have been hearing this term. Information technology also is quite a old term. And cognition refers to the ability to use the mind and to think rationally. So this NBIC, and in that you can add A also, NBICA. This is going to be the order of the day. And along with this, the futuristic courses or the concepts include artificial intelligence, robotics, nanotechnology, nanobiotechnology, 3D printing, big data, and the omics. Right. So with this concept, we'll now move on to the next part of the slide. A quick recollection of what I've already explained to you. So STEM learning. STEM learning involves integration of scientific inquiry. What does this inquiry mean? Meaning you have to be inquisitive. The minds of the students need to be very, very sharp and always willing to learn. You should have the mind of what Alexander Fleming or Archimedes or Newton had during their discoveries. The next is technological literacy. So you learn how every technological aspect works and you try to develop that in large scale. It could be a manufacturing of 100 tablets, and it could be extrapolated to millions of tablets in a large scale, right? And then you have to use the concept of mathematical thinking and ultimately the engineering design concept. So this together is called as STEM learning, and this is used in the community and it is practiced in the community for societal applications. So next is an integration. We'll just go a little fast because I already explained to you. The A stands for art, creativity, and the ability to communicate and write the scientific presentations. So this is what is called as STEAM concept. So now coming to the basic sciences, life sciences. Excuse me, please mute those who are having the phone. There's a lot of disturbance, please. Yeah, with regard to...
Kindly mute others, please be on mute. Those who are joining now, please mute yourself.
Yeah, thank you, madam.
Yes, sir, you can proceed, sir.
Yeah, with regard to all our medical and paramedical professions, we are all having life sciences as a basic study. So based on life sciences, what are the other integrating disciplines? We have one is what is called as mathematical modeling, which is very, very useful for epidemiologists, right? I'm sure the medical epidemiologists in the university and all other places are using this mathematical modeling concept. And then you have got an evolving field called as systems biology. And you have other keywords in the entire slide. You can just have the students can look at the words which I have highlighted so that you don't need to get carried away by a lot of written matter there. The second one is the importance of biomaterials, where nanotechnology has a lot of role to play, where you have nanotechnology in the targeting gene therapy as well as re-engineering new tissues, right? Organ on chip, and all these techniques are related to evolution of biomaterials. And then you have cells for use in tissue regeneration, which I told you about regenerative medicine, a whole branch of medical field is developing so much. And then you have another important field which is called as biomolecular research. So here it is all about the pharmaceutical discovery process. Now you have pharmaceuticals which could refer to small molecule drugs, you could have peptide drugs, and you could have protein-based drugs. So the peptides and the proteins together are called as biomolecules. And in this biomolecule research, you have got the systems biology, which plays a very, very important role, about which we'll see later during the part of the presentation. Right, again, little bit of more explanation on synthetic biology, which integrates computer science with genetics, molecular pharmacology, or molecular biology, and it is involved in constructing new biological components and systems. Right, so here it is you are able to incorporate these systems which are prepared in small scale into larger systems and see how they function. So this is where the function of an engineer comes into picture, right? And if you see beautifully, there have been a lot of low-cost diagnostics which have been developed using this synthetic biology concept, especially for Ebola. In various pandemics, you are able to develop almost 20 different sensors for Ebola within a day. So this shows how much the synthetic biology has been able to penetrate. So next, another very, very important field of integrated learning is the microfluidics. Now, microfluidics integrates life sciences, physical sciences, and the engineering sciences, right? If you can call it as a science. Right now, it's all together. There's nothing which is really bifurcating. Indian Institute of Science has started engineering courses, and IITs have started science courses. So that is how today's world convergence is happening, right? So convergence for biomedical research involves integration of life sciences, physical sciences, and engineering. This is here where microfluidic systems have been developing very, very rapidly. So what is so different about microfluidics? Basically, they are different from normal flows, right? They are almost like a laminar airflow hood where all the air which is flowing out is in laminar direction. So this gives microfluidic systems very, very great properties with which a lot of research is being carried out. And next, an important aspect or development of new application of microfluidics is the development of point of care diagnostics. This point of care diagnostics has helped the scientists to develop rapid diagnostic tools. Even in the case of COVID-19 and other infectious diseases, you use this microfluidic technology and you come out with newer techniques. Right? The simple example is the kit which is used for testing pregnancy, which is based on the urine test, and you've got the human gonadotropin, which is identified in the strip, right? So they are called as self-contained or standalone microfluidic systems. You need to apply the basics of physics, basics of biomedical engineering, all these things towards benefiting the society. So here, if you see, the important keywords in microfluidics are the spatio-temporal precision with which these systems work, and they are able to simulate similar physiological conditions as what a cell experiences inside the body. This is the beauty of microfluidics. You're able to carry out the entire research in small scale, in micro scale, and you're able to simulate the same conditions which happens inside the body. So spatio-temporal precision and the similar physiological fashions the cells are exposed to is the basic big advantage of microfluidics. And using microfluidics, we have been able to come out with models which simulate blood vessels, muscles, bones, liver, brain, gut, and kidney. Most of the organs have been modeled using microfluidics. So you can see, though we have not been so familiar with this term microfluidics, you can see how much of a penetration this microfluidics has done in our own medical and paramedical field. And the next important thing is, again, overlapping with pharmaceutical field, that is toxicity screening of drugs. Here you see now the days of screening with animals is getting outdated. So you've gone in for more of microfluidic models where you can use cell lines and you can go in for toxicity screening of drugs, right? And then next, furthermore, I think I'll skip on. The most important advantage, right, of microfluidics is the ability to encapsulate a single cell in droplet. As I told you, you're able to study the behavioral pattern of every cell and able to study how the cell changes with changing environment. So we talk of homeostasis and all these things. So using microfluidics, carry out the entire work with in small scale without wasting much of reagents and all those things. Economically, you're able to do it. You're able to do it at a very, very fast rate. High throughput screening can be adopted, and you can study single cell in droplet forms. So this is one of the most important advantage of microfluidics. I hope I've made it clear for undergraduate students. I know that it is going to be a little difficult, it may be a little heavy, but just try to go through, and any basic knowledge on microfluidics will help you know how much it is penetrating into a medical field also, right? So as I told you, you've got what is the beauty of analyzing every cell instead of studying cells in population. If you're able to study individual cells, you get a better picture of what exactly is happening. This because every cell behaves differently. This is especially true in when you study tumor progression at every phases of cell cycle during tumor progression you might have studied, right? So there you can beautifully study using this microfluidic system. So stem cell differentiation as well as host pathogen interactions, you can use this microfluidic concepts. I hope I have been able to give you some insight to microfluidic applications in medical research. Okay, so next, this is a beautiful diagrammatic representation of how every organ has been represented in a small chip. It's something like a SIM card, right? You've got a heart with the cardiac muscles and cardiac tissues which are incorporated in the chip. So this is a beautiful example of a organ on a chip. Next, very, very important concept again, of which we have been hearing for the last 2 decades, that is nanotechnology, right? So here, what does this integrate? It integrates again physics, just like your microfluidics. Physics is the basis. Material sciences, about all your polymers, nylon and biodegradable, biocompatible polymers. All these are related to material sciences and biotechnology, of course. So that's why this combination is so vintage. Nanotechnology itself has got so much of penetration. Now, when this nanotechnology combines with biotechnology, it becomes nanobiotechnology, and the implications are several fold. So as you all know, by now you are all familiar, nanotechnology is understanding the and controlling matter in the nanometer range, that is 1 to 100 nanometer range, right? And the nanomedicine, this is another nano prefix is available in so many terms: nanotechnology, nanomedicine, precision medicine, nano spheres, nano drops, so many things you use, right? Nanomedicine is used to refer to use of precisely engineered materials to develop novel therapeutic and diagnostic modalities. So nanomedicine is used both in diagnosis and for therapies, especially in cancer therapy. Nanotechnology has been phenomenally growing. Right? A typical example is nanocurcumin. Nanocurcumin, if you put the keyword in the Google search, you will find millions of research work being carried out on nanocurcumin. You will find almost 10,000 times or few thousand times the dose of curcumin can be reduced when it is formulated as a nanocurcumin form. Right. So brief introduction about basics of nanotechnology. Right. What exactly is nanotechnology? If you see the diagram, it may be a little confusing for you, but just see that the one which is center, it is a diamond, and the one at the top is the feather, and the one at the bottom is the spacecraft. So carbon, you all know carbon is the basic elemental makeup of a diamond, right? So you can use this carbon, and by nanotechnology, you can create materials which are as light as feather and which can be even used in spacecrafts. Because if you're going to create a very, very heavy metal or metal object, it's going to be very difficult for you to launch the spacecraft. So nanotechnology has been able to create materials which are as light as a feather, peacock feather, and which are can be used in spacecrafts which are so rigid, which are so strong at the same time. So light as well as strong. And as I told you, in cancer diagnosis, you can use the traditional photodynamic therapy, which is used through nanotechnology. Right now, this is one slide. You will have a lot of articles on nanotechnology in pharmaceutical application. This is just a brief slide on what are the various areas where it is covering. Right, just like your computers, which are used in so many fields, the penetration of nanotechnology is almost in every field. And on the top, you can see the various types of nanoparticles, which can be called as dendrimers, liposomes, quantum dots, fluorine tubes, nanospheres, carbon nanotubes, and all these things. So this is all the classification of the various nanosystems, and the various applications could include in gene therapy, in cancer therapy, diagnostic, as well as in therapeutics, in genetic disorders like thalassemia and other myasthenia gravis and so many other diseases, as well as in schizophrenia and all other CNS related disorders. You can use nanotechnology. We'll see over the pharmacological and cardiovascular diseases, also in hypertension, how nanotechnology is used. So this is the, in short, what are the various areas where nanotechnology is penetrating, right? So you can see how the futuristic nanotechnology is going to be. By 2024, the nanotech is expected to exceed $125 billion. And by 2025, the smart pill, global smart pill market will reach about $650 million, right? I'll show you later how what is the exact thing of a ingestible capsule, where the sensor is incorporated in the capsule and you're using it for studying the various parts of the intestine, as well as for Crohn's disease and other diseases. So this is how nanomedicine is really penetrating. And one example, classical example of the application of nanotechnology. This is one of the first examples where it has been used in drug screening. So this is actually a novel work carried out at the London Center for Nanotechnology. If you just Google it, it will get this beautiful diagrammatic representation of this. I'll briefly explain to you what it is. So this London Center for Nanotechnology people, the scientists have developed a novel nanomechanical approach to investigate vancomycin. Vancomycin, I'm sure the medical and paramedical people will be familiar. It is a reserve antibiotic, and today's world of multi-drug resistant organisms, vancomycin is almost the only resort or the final antibiotic which is to be used. So they have used, scientists have used this cantilever arrays for studying using nanotechnology how vancomycin acts. As you all know, similar to your cephalosporins and beta-lactam antibiotics, vancomycin also acts on the cell wall of the bacteria, right? So they have used this cantilever arrays for studying how vancomycin is able to act on the cell wall of the bacteria. So the principle is based on cantilever arrays, which are coated with a mucopeptide, and the antibiotic attaches to the surface, and this attachment results in the bending of the cantilever, and that is what is shown in this particular diagram. So the representation which clearly shows how much of bending of the cantilever happens, and this indicates that the antibiotic has acted on the particular cell surface or on the cell wall, right? So this has been used to not only study the mechanism, but also to differentiate between resistant and non-resistant strains. So it's very, very important to know epidemiologically what is the level of resistance spreading in a particular population. So the physicians who are going to be prescribing the drugs based on the antibiogram of the particular microorganism will have to know whether the organism prevalent in that particular society is a resistant microorganism, especially in tuberculosis like diseases, or a non-resistant organism for which you can use conventional normal antibiotics. If it is a resistant organism, you immediately will have to go for moderate and very, very penetrative antibiotics. So this cantilever based approach can be used to differentiate between resistant and non-resistant organisms. And if you see the beauty of how the resistance develops, we are talking of convergence of all these technologies, but you can see how God has created the microorganism. Just these are called as super microorganisms, which are multi-drug resistance, are also called as superbugs. MRSA, methicillin resistant staph aureus, is a classical example of a superbug. And you can see how it is able to adapt itself and it becomes resistant to the antibiotic. So what is this mechanism exactly? It is a simple mutation that deletes a single hydrogen bond from the cell wall of the particular structure, making the bacteria, making the superbug 1,000 times more resistant to the antibiotic. So this is how these superbugs have developed resistance, and it is evolving. It is a day-to-day changing phenomena about which the pharmacists as well as the biotechnologists and the physician need to have awareness. Right? So this is with regard to the nanotechnology and the cantilever approach. We'll now move on to the next integration. That is integration in the next field of pharmacy, that is pharmacy practice. In the case of pharmacy practice, it is a new term which is now evolving that is called as health technology. Health technology, as per WHO, is defined as the application of organized knowledge and skills in the form of devices, medicines, vaccines, procedures, and systems developed to solve a health problem and improve quality of life. So it could be a health technology could be developed by a retail pharmacist, by a community pharmacist, by a hospital pharmacist, or any other pharmacist who's practicing the profession of pharmacy. Right? Of course, in developed countries, technology has developed so much, and they are in sync with what is happening. But probably in a country like India, we need to catch up with the technological development and use it as and when it is required. Right? Especially in COVID like times, we have seen how automated dispensing and telemedicine and telepharmacy has really worked wonders, and India has been able to manage this COVID pandemic spread so effectively. This is all due to telemedicine on all these so technological pharmacy practice services, right? So you have computers which are integral part through which the pharmacist is able to work out, and you have the software which can be recorded in the computer, and all the interventions according to the patient's details. If a patient has got penicillin allergy, immediately the pharmacist or the doctor is known about it, and he avoids using penicillins and goes in for the next line of drugs. Similarly, if you see face-to-face consultation is very, very important in pharmacy practice. It's always better to sit in front of the patient and then counsel the patient. But today's COVID times, it is not always possible, and we may have to skip all these things. Right? Next is with regard to pharmacy practice, advanced robotics, right? Use of internet, and then wherever it is, it may not be always feasible. This particular use of technology in pharmacy practice in a developing country may not be always possible because of cost constraints. Right? So it is wherever possible, please try to adopt. And in the truest sense, technology is not just manufactured objects and technological procedures, but also incorporates human inputs and clearly defined operational processes. So this is with regard to pharmacy practice. And you've got another two more terms which I came across during the preparation, that is general purpose technologies and technology enabled pharmacy. So this technology enabled pharmacy is what I said, using robotics or using convection belts in a hospital setup to dispense the pharmacy or the medicines to various wards without any human interface. You don't need to use retail pharmacists to do the job. You can use conveyor belts and use technology to dispense the medicines, bar-coded formulations. All these things help in technologically driven pharmacy practice services. And pharmacists have always been technologists, and their work has led to the development of an untold number of innovations benefiting the trade of pharmacy and the life of patients. So ultimate aim is to improve the quality of life of the patients, right? And then inseparable word again, today's buzzword, we call it as big data. What do you mean by big data? You collect as much of information possible from as many patients, and a computer integrates all these data, and you make out or you develop some meaning to the particular data. Like, for example, I told you about the spread of antimicrobial resistance. So within a particular population, in and around Coimbatore or in and around Tamil Nadu, what are the organisms which are resistant spreading? It may be different from what is spreading in another country. So this depends upon all the environmental factors, and this is where big data can really be useful for pharmacists as well as for doctors, right? So epidemiologists, again, I told you about mathematical modeling and all these things. Epidemiologists will greatly benefit due to this big data and other computer-aided methods of pharmacy practice. So this completes pharmacy practice integration. And next is with regard to pharmacological integration. So when you say pharmacology, it is about diseases and how drugs act and what are the various drug of choices for various conditions. So two important diseases or three important conditions I have chosen. One is my atherosclerosis, very, very important, resulting in heart attacks and ischemic heart disease and so many other things. So I'm not going to go into the pathophysiology of the atherosclerosis. You all know it is due to excessive accumulation of lipids, and the entire blood vessels becomes narrow, and this results in either thrombus formation or even sometimes embolus, which starts circulating. Accordingly, it is called as a pulmonary thromboembolus or cerebral thromboembolus or whatever it is. And here, nanoparticles using either nutraceuticals or other pharmaceutical drugs which are used in atherosclerosis, right? You've got statins and other agents which can be used. They have been formulated as nanotech based drugs formulations. And here they have found the half-life of these drugs have been increased several folds. So this is the advantage of, so anybody who is working on nanotechnology and on atherosclerosis can probably combine these two things, and you can monitor how this is effective. So next is with regard to hypertension as well as in pulmonary hypertension. I just quickly go through because still there are a few slides to be covered. Right here in this case, with regard to hypertension, you have the sartans. The olmesartan has been formulated as a nano emulsion, and most important advantage with nanotized formulations are the drastic reduction in the dose. So when you're able to reduce the dose, automatically the side effects and the adverse effects can be accordingly reduced. Particularly in the case of pulmonary hypertension, you have busantan, which is a selective and competitive endothelin receptor antagonist. You've got several groups of drugs which can be used in pulmonary hypertension, of which one is the competitive endothelin receptor antagonist, where bosentan has been taken as a model drug, formulated into a nanoparticle, and they have found its solubility has been increased 7 times compared to normal and bosentan. Right? Similarly, I told you about curcumin and nanocurcumin. The solubility is a major problem with regard to formulation of a curcumin, which can be overcome by formulating it as a nanocurcumin. Right? So this is with regard to your pharmacology. Next is in fields of bioinformatics, almost overlapping with biotechnology. Right? Again, these are all difficult concepts for undergraduate, I understand, but it's high time we start familiarizing ourselves with all these topics. Right? So in bioinformatics, how integration happens is through a series of biological databases, of which KEGG is one of the important biological database, Kyoto Encyclopedia of Genes and genomes. I have also given you the hyperlink. What are the things which it can give about? There is a wealth of information which can be collected through this KEG, right? This Kyoto Encyclopedia of Genes and Genomes. It helps us to understand the entire pathway of diseases, what happens, what are the biochemical changes happening, everything. It's a established database, biological database, which has been compiled using published data, right? There is no single information which is false. The entire information. So very, very important. Today's world of internet, you need to know where from you have taken the information. Today, students come out with so much of information, but they are not able to filter out. So the most important thing is how you filter out. So this KEG software is a biological database which contains all the details regarding the complete information on the genes, the protein-based things, and how the biochemical things happen in the cell. Everything is it's a biological database, right? I've given you the hyperlink also. Those who are interested can probably in the question hour you can ask few things after that, right? So I told you about the information. It's 100% reliable because it is all compiled from the published data around the world. But the thing is, the data from all the microorganisms are not currently available. This is being updated. So this is the home page of this KEG biological database, and this is what information which it can give. So anything about related to the disease, related to the metabolic pathways, as well as the pro-drugs and all these things, you can study it under this particular database. Currently, there are about 19 KEG databases which are updated, right? So this is with regard to bioinformatics. Then next, extending with biotechnology, right? I'll try to be a little short. I know I've been quite elaborate on so many things and a lot of new concepts, but this again is a very, very important topic called as peptide therapeutics. Maybe for pharmaceutical students, pharmacology students, right? Because out of the top 10 or 20 selling pharmaceuticals worldwide, you will find almost 60 to 70% of the drugs belong to this category. I don't need to stress more about the importance of peptide therapeutics. I told you that pharmaceuticals could be small molecule drugs, peptide drugs, or protein-based drugs. So these peptides and proteins together, they are called as peptide therapeutics, because most of the peptides are natural macromolecules, are natural receptors or neurotransmitters. All the naturally present substances in the body which catalyze or which control the entire biological process are all protein based. So that is the reason why this protein therapeutics or peptide therapeutics is gaining so much of importance, right? I'll just highlight the important things here. Peptide therapeutics are produced mainly in a biological medium utilizing genetically modified living systems. So here when you say genetically modified living systems, I am not talking of rDNA technology or enzymes which are modified, like you got the immobilized enzymes. All these things are separate topics on their own. I cannot cover that during our topic now, right? I'm just giving you an overall view, right? Here peptide therapeutics, you are using mainly they are produced mainly in a biological medium utilizing genetically modified living systems, which are recombinant microorganisms, right? And their synthesis is influenced by multiple parameters during the manufacturing process. So this may lead to changes in the 3D structure of the biotherapeutic so as to impact the quality, safety, and efficacy of the therapeutics. So when you study about peptide therapeutics, you will understand whatever I have told you, because the three-dimensional structure of a peptide molecule, primary structure, secondary structure, tertiary, and quaternary structure is so important for its biological activity. That is what it's called as peptide-peptide or protein-protein interaction. Drug-receptor interaction is all based on three-dimensional interaction. That is the reason why peptide therapeutics is gaining so much of importance. So quickly, I'll skip off. I'll just tell you some important concepts again here. It is called as antibody drug conjugate, of which there are only about a few products which are approved. I think there are currently only 2 antibody drug conjugates which are available in market, and most of these are used in cancer chemotherapy. Right? Those who are working on cancer chemotherapy, I'm sure you would have come across this antibody drug conjugates, and it's a beautiful diagrammatic representation of how this works. So the payload is nothing but the drug which you use for treating cancer. It could be a anti-cancer drug, or it could be a transcription factor, or it could be a nucleotide, or it could be a non-radioactive effector. Because as you know, cancer can be treated surgically or by medically or by using radiation. So any one of these concepts, three methods of treating cancer. So the payload is nothing but the drug which is used, right? So the drug is attached to a carrier, and for attaching the drug to the carrier, you require a linker, and the linker could be a non-cleavable linker or a cleavable linker, depending upon what type of response it has to be used to open the drug from the carrier. So the cleavage will result in opening of the drug from the carrier bond, and it will release the drug for the anti-cancer property. So the main advantage of the antibody drug conjugate is you can target this antibody drug conjugate to the cancer tissue, and you can have a lot of selectivity in action. Side effects can be drastically reduced. You all know that anti-cancer drugs are highly toxic. So this is the single most advantage of antibody drug conjugates. Right now, we are almost coming to the end of the talk. Right here, I again highlighted the important keywords: converging technologies and healthcare. We have seen a lot of things starting from systems biology, synthetic biology, microfluidics, nanotechnology, bioinformatics, peptide-based therapeutics, all these things pharmacologically, how they are used, right? All these integration, how it is used, all these things we have seen. In pharmacy practice care, how it is used. So what exactly happens? Convergence in healthcare is a guiding philosophy for future medicine. Let this be very clear. Future medicine is going to be guided by convergence in healthcare. And here, it is not a single person who can really give the output. It requires participation from a physicist. You may wonder, a medical or a pharmacist or a nurse may wonder what is the role of a physicist. But today's world, it's all changing. A physics person can interact with a doctor and come out with a lot of benefits for the society. That's what I meant by guiding philosophy for future medicine. Integration of physics, mathematics, engineering, computing. Like computer is integral part. Chemistry, chemical sciences, and life sciences. And the four key areas are biosensors, where a suitable biomarker is actually identified, and you go in for small scale or you do it in like a microfluidic involved technology. You can go in for development of biosensors, or you can go in for interventions or term like precision medicine, which is again the benefit of converging technologies, and you can develop healthcare platforms and population health tools. And the converging technologies aims to understand the basis of life and disease processes and use this to develop revolutionary new molecular and cellular therapies and diagnostics, instrumentation and data analysis methods for health applications. Right. On one more slide on convergence and healthcare, the knowledge, society, technology, and natural systems are coherent. So what I mean is they're all interrelated. Right? Knowledge, society, technology, and natural systems are interrelated, and they have to be studied together. Convergence provides an unifying strategy that can be applied to all systems like evolving neural networks. So if you study the brain, that's why they say the brain is one of the most widely studied organ. It's very, very difficult for in one generation, you cannot study the entire brain because the network which is happening inside the brain, the neuronal connectivity is so great, right? So convergence provides unifying strategy that can be applied to all systems, right? And here you've got three keywords: nanotechnology, digital technology, and artificial intelligence. This is what I said in the beginning of the slide. NBICA: nanotechnology, biotechnology, information technology, and your cognitive sciences, ability to conceptualize, ability to comprehend, ability to assimilate all the data, and ability to translate into real output. And along with that, you need to communicate using art and communication. So this deep integration of the five basic science and technology disciplines will really go a long way in improving the healthcare, right? Then a few high technological terms which you can come across, right? Like pill cam. I told you about how the camera can be incorporated into the capsule, and you can use it for studying the gastrointestinal tract. Similarly, to monitor whether a patient has taken the drug or not, you can use this dose tracking pills, and you can also use vibrant capsules, especially for you don't need to use an irritant laxative right? Like senna or any other thing, castor oil. You can just go in for this vibrant capsule, which will just vibrate inside the stomach and it will induce defecation without need of any drug at all. So this is one of the technologically advanced formulations which are available. Then you have what is called as an atmo gas. What is this atmo gas? A lot of importance today is being given to the natural gut microbiome. The intestinal microbiome is an indicator of how healthy a person is, right? So here, if you see the levels of dissolved oxygen, hydrogen, and carbon dioxide, if you can monitor, this will help you to identify whether a patient is physically and clinically all right or not. Otherwise, you can say that he is abnormal. So this is how this atmo gas capsule has been able to identify the levels of oxygen, hydrogen, and carbon dioxide in the gastrointestinal tract, and you come to a scientist comes to a conclusion as to what is the health status of a individual. So this is another capsule, and then you have, so this will resemble a TV antenna which you have in your for providing Wi-Fi connection in your house. So this has almost come replicated in a capsule also. This is called as a MIT capsule, that is Massachusetts Institute of Technology people have come out with a smart sensor capsule. So it has got a Bluetooth connection and all those things, and the data can be integrated. And then you also have smartphone microscopes. What I'm trying to tell you is not hypothetical. These are all going to happen within the next decade or so, right? Here you have used smartphone microscopes. So instead of just wasting our time on WhatsApping, on probably putting our status and other things, I think you can use these smartphone microscopes to come out with something novel, right? A smartphone application that can detect nanoscale particles of norovirus in the water. So all the mechanism and other things has been illustrated there. And then you have got, I think probably the last slide, you have nanopatch vaccines. Vaccines, I'm sure you all know, are the during this pandemic, COVID vaccine has been instrumental in our country being able to export so much of vaccines, and how we have been able to overcome this pandemic, right? Here, vaccines, the main problem is most of them are given as injection. So if you can overcome that problem and give it as a nano patch, so nanotechnology is also used, and convenience of administering through the skin results in these vaccines being administered. Similarly, you have smart bandages, where depending upon the infection inside the body, the drug will release the antibiotic, and it will have a sensor. Depending on the infection severity, the drug release can be controlled. Right? So this is all some of the futuristic technological advances, and this is still more fascinating. I told you about. I'm sorry. Yeah, this is an animation actually, just a 30-second animation. This is so wonderfully taken from the Google only. Right? This is called as clarity. This is called as clarity. This is a three-dimensional image of the brain. This is a three-dimensional revolving image of the brain, and slowly you can start observing the neurons. And now those lines which you see are all the dendrites on the neuron. So you can imagine how it is. Instead of explaining using a diagram in the blackboard, you can see it in the microscope and the computer itself. These are all the dendrites of the neurons, and the entire brain can be studied in the three-dimensional way. So you can use this particular study for identifying what exactly is happening inside every cell of the brain, and it will go a great long way in coming out for remedy for Alzheimer's or myasthenia gravis or any other central nervous system disorders. Right? There are quite a bit. So coming to the last slide of our presentation on conclusions of integrated disciplines for improved healthcare. So what exactly is the keyword here? It is innovation. Innovation in early detection, disease diagnosis, treatment monitoring, and precision medicine. All these things have to be innovatively used, and this is the real need of the hour. And anchoring these developments, having the following the principles of ethics, policy, and society will enable acceptable and sustainable solutions to overcome the challenges of the society which we are currently facing. And all these challenges can be overcome only through convergence and interdisciplinary workout. So this is what I'm sure it will be very difficult for me to really go about explaining to you about nanotechnology and so many other information. Rather, I thought it would be better if I give you a broad perspective of where all these disciplines are able to penetrate. So the last thank you slide, if you see this clearly, this is the representation model which is kept in the Bangkok airport. So I am sure you all know it's a Muslim dominated country, but still they have taken the necessary care to install a character or a incident which is more related to Hindu mythology. This is the churning of the nectar of ocean, right? Ocean of nectar with the snake, and one side you have the asuras and the devas who are churning the amrit. So amrit, is the magic potion. So whether the whole concept of medical team is to come out with a magic drug or a magic potion wherein the society can be totally disease free. And what is going to be the magic potion? You need to churn with the pharmacists, with the physicists, with the biotechnologists, with the nanotechnologists, with the computer technologists, with all the scientists integrating together and coming out with a interdisciplinary approach to create a healthy society. So this is what is in short the overall convergence of various disciplines. And before I really conclude, I'd like to place my humble thanks to our Honorable Vice Chancellor, to the Registrar, and all the other officials of the Tamil Nadu Dr. M.G.R. Medical University, and of course my principal, my vice principal, and my managing trustee, and all the faculty and students who have helped me during the course of the presentation. I would also acknowledge the thanks of Dr. Grace Ratnam and Dr. Sampath Kumar, the moderators of this particular talk, and all the students who have given a patient listening. I only hope that you have at least grabbed the essence of what has been covered, and you will be able to interact further and make use of whatever I have been able to share during this short period of time. Thank you so much, and thank you all. Thank you.
Thank you, Dr. Krishnan, for the very lucid presentation and for seamlessly bringing together so many disciplines. The session will be now open for any questions. All the participants, you can please ask your questions, introduce yourself, ask your question, and then please remember to mute yourself.
Hello, sir.
Yes, sir.
Hello, sir. Good afternoon.
Good afternoon.
Sir, how the converging technologies will change the future of healthcare?
That's what I've been explaining throughout the 1 hour, no? Both in diagnosis as well as in therapeutics of cancer-like diseases, brain disorders. Right? You need to work together. Supposing you want to come out with a new system, this thing for using microfluidics, you need to be a physicist. You should know basics of physics. You should know basics of biomedical engineering. You should know how it can be used for diagnosis of, say, pregnancy. You need to come out with a microfluidic based test for pregnancy, or you take COVID itself for that matter. You need to come out with a new test for COVID. You can use all these concepts, microfluidic. Now, recently, I think there was a latest technique which was able to identify the virus within 1 day or even few hours, right? Rather than your PCR-based identification, if you can use all these converging technologies and within a very, very short period of time, if you're able to diagnose a particular disease or the spread of a particular pandemic, it really helps in healthcare monitoring, isn't it?
Okay, sir. Thank you.
Yeah.
Thank you. So one more question, how it can be focused on academic side, educational institution?
Yeah, educational institution. Yeah, this is a very, very important question. See, we need to assess ourselves where we stand according to the infrastructure facilities which we have. Okay, so we may not have a lab which is available in a developed country like US or UK or Australia or any other place. So according to the lab constraints, that's where you need to integrate and find out what type of problem you need to sit together with your guide, right? Which area fascinates you? you should have a clear goal. Next 4 years, 5 years, what you want to do. So what you study in B Farm is only a overall picture. The beauty of the B Farm profession is whatever I've explained to you, it is not very far away from B Pharmacy. Okay. A B Farm can very well equip himself in related to microfluidics. He can come out with a new technique or a new it's called as point of care technique or organ on a chip. You can come out with a new organ in a small chip. To this extent, you can work. So only thing is you need to identify what facilities your lab has got. For example, I'll tell you, in our lab, it has taken almost 5 to 10 years for me to establish genetic profiling. So we are carrying out genotyping of microorganisms which are resistant. So we have worked on gram-negative bacteria, we have worked on MRSA, and over 5 years, PG students have been able to develop a method by which genetically they identify in and around Coimbatore, these organisms have been found to be resistant to the antibiotic. So you need to know how this PCR technique can be used, how you can use gel documentation, and how this genes can be identified. I'm not talking of gene sequencing and all high-tech methods. I'm talking of basic techniques which can be carried out in the lab. Like, for example, nanoparticle formulation. You identify your problem in your lab, whether you have got facilities in your Sutix lab to prepare a nanotized drug. You try to do that, identify your clinical problem, which disease you're working out. Basics should be clear. And then depending on the infrastructure, you need to choose your further this thing. So I told you, in our lab, we have been doing resistant genotyping of resistant microorganisms. We have been able to establish MRSA and gram-negative bacteria, and we have even correlated bioinformatic tools. Using bioinformatic tools, we have correlated with the resistant strains. Clinically, we will collect the strains from the hospital. We'll do the antibiogram, and then we'll correlate it with the bioinformatics data. So that is how you expand. What I have talked so far cannot be established just overnight. It will take at least few years and few years of planning to establish this level of work. That's why biotechnology is still nascent in our country, right? You need to establish centers of excellence and you need to pursue your goals accordingly. I hope it's clear, because what you're asking is more hypothetical. I understand, but I'll be happy to interact with you more, right? Maybe you can email me, or maybe later you can talk to me even.
Yes, sir. Yes, sir. You are clearly explained about the research. So maximum each and every research we have to implement innovation. With the help of innovation and also interdisciplinary research, we can achieve the goal.
Very true, sir. And startups, you heard of startups,
yes,
isn't it?
Yes,
and
sir, yes, sir.
Tinkering labs. Today's this thing we have seen schools also being changed into. You got what are called as Atal labs.
Yes, sir.
In remembrance of Atal Bihari Vajpayee, you got those tinkering labs where school children are being exposed to high-tech exposure to scientific knowledge. Similarly, you have hackathons in engineering discipline, hackathons. And I would request our students to please go to TED Talks, right? I'll explain to you what exactly. If you go through TED Talks, if you listen to TED Talks, you just type the keyword, what you're interested in, and you download. It will be only 10 minutes presentation, right? We are hearing so many things on YouTube's best things. I think TED Talks will be really, really useful to you. Right? Just see what fascinates you. Type the keyword in tedtalks.com, and then you will come across excellent inspirational words of knowledge, right?
Yes.
So, as sir said, innovation is the key.
Yes, sir. And also, before starting a research, we have to problem. We have to start a problem, and with the help of identify the problem, and we can go for a solution.
Yes.
So then only it will be innovative.
Yes, sir. Yes, sir. That's called triangulation in research, sir.
Yes, sir.
That concept is called as triangulation in research.
And also, I suggested our VC madam to implement this innovation and design thinking and critical thinking and this concept to our syllabus. It will be very useful for students, young generation. We can motivate in the innovation technologies.
Absolutely, absolutely. I think probably we could even think of probably the people in the board of studies and this thing could think of including innovative topics in the electives.
Yes, sir. And also the national education policy also insists we have to more concentrate on innovation.
Hello, sir. Audible?
Yes, continue.
I think we have Ms. Sampath is cut, I think.
Hello.
Yeah.
I'm here, sir. I am here, sir.
Okay, sir. Okay.
Sir.
Yes, sir.
Continue.
Sir, regarding NBIC, nanotechnology, biotechnology, information technology, cognitive science.
Yes, sir.
Sir, I have seen that one information. With your permission, can I read that information?
Please. I have not. Please read. No problem.
Yeah, if the cognitive scientist can think it, the nano people can build it.
Excellent.
The bio people can implement it.
Yes.
And the IT people can monitor and control it.
Very good. Excellent.
Because these are the 4 research challenges described in the NBIC.
Fantastic. Very nice. That's what I said. You have a wealth of information in the internet, but as students should know what to filter it out and how. So you have to hear from the horse's mouth who is telling what information that has to be taken. Right? When I say this is a cantilever and the cantilever concept can be used to study mechanism of action of drugs, immediately you should go into London Center for Nanotechnology, and you should see how beautifully they have invented that cantilever for studying penicillin mechanism of action. That will inspire you. Thank you, sir.
Thank you, sir. Thank you very much for your attention.
Thank you. So as they say, after lecture, no questions indicates 2 things.
Sir, I will ask, sir.
Yes, sir, please.
So many research are going on technology, what you explained, more than 10 technologies. So, but if you're going for outcome, only 1% of research is going to be succeed and coming to market. So, yes, what is the backlog of that mission?
Oh yeah, fantastic question. I'll try to explain of giving you an example of one information which I have collected probably through my sources. Right, this is what is happening in one of the developed universities abroad. Right, what we all are unaware of what is exactly happening in China. Most of us, at least, we have not had exposure to Chinese education and all those things. But I have heard one of the important first-hand information on what exactly is happening through one of the reliable sources. Right there, what you see, there was one of the presentations in a developed university, and people from China, right, they came to market one chip. It's something related to nanotechnology, nano and biomedical research. So that one chip, they came to market, and they are coming to another top university, one of the top 20 universities in the world. And if you see the data which they have collected is through use of several supercomputers. Probably every small university in China is equipped with a supercomputer, and the population, they have the strength of population. So the entire population, they collect all the data, and the data is assimilated in the computer, and that data is incorporated, and they say that one chip contains data related to, say, multi-drug resistant organism. Say, for example, Mycobacterium tuberculosis. This is the complete information of Mycobacterium tuberculosis organism present in China. Like that, they are coming out with a microchip, and that microchip, it costs in lakhs, it seems. Right. So what I'm trying to tell you is, it is the ability to use data, assimilate the data, yes, and for that you require infrastructure. So unless you have infrastructure, and every lab and every colleges, or at least every universities in our state and country should be having a supercomputer. But to my knowledge, I think even states may not have supercomputers. So that is what is integration is more important. Then yes, automatically outcomes will automatically follow. So as I told you, you can go in for startups, or probably you can even interact with governmental organizations and patent your product, right? Product patent or process patenting can be done, and you can gain overnight publicity. So this is still hypothetical, I know, but I'm sure this talk would inspire you to reach further goals.
In continuation of your just the question asked, after so much research, these nanosystems like liposomes, all that, if you see the approved and marketed, mostly it is for treatment of cancer.
Yes,
It is
madam.
already in the market and approved also. For what other conditions, what other diseases or research is going on, and what will most probably come out?
Almost everything, madam. In fact, I had explained to you, no, madam. In fact, few diseases I have not taken up. You can, based on CNS, the research is basically based on areas where more developed countries can fund. So funding is a major constraint. Nobody works on malaria. Nobody works on Ebola. Nobody works on Kala Azar, though it is more prevalent in developing countries. So basically, what I found, I'll just probably share with you one more slide, and that so that I can, yeah, this slide. Are you able to see, madam, this slide?
Yes.
Yeah. The most of the material which I have collected for those people who have found this seminar useful, you can see the third reference, that is Convergence of Health 2016. That is a 80-page report given by Massachusetts Institute of Technology. Now, this is an university which is #1 ranked in both medical as well as engineering fields. Okay, Massachusetts Institute of Technology. So you can imagine the level of research which is happening there. And this is a 80-page report which they have shared. And this is shared to the entire world. So Convergence of Health. So here they have beautifully given individually where all it is useful. It is useful in, of course, cancer being the most important. Similarly, you have got in central nervous system conditions, like for example, schizophrenia, in myasthenia gravis, in various other cerebral palsy, right? Similarly, in autonomic nervous system disorders, cardiovascular disorders, atherosclerosis, multiple ischemia, or multiple sclerosis, or so it goes on, genetic disorders, gene therapy. This is almost an ocean of knowledge which I have got. So it's very difficult to concisely tell what exactly is can be the real output. It will require at least
it's been successful for cancer to a large extent, and for others will be going on. I have now one question in the chat box from Dr. P. Amuda.
Yes,
she
madam.
says, I would like to know if there's any software for peptide therapeutics and 3D brain.
Yes, madam. Clarity is the this thing for the 3D brain. That if you go into see Clarity, this thing you can get details. If you want, I can share that also related to that. 1 second. And the other one was related to peptide therapeutics. So with regard to peptide therapeutics, again, bioinformatic tools, I told you about 19 different tools which are assimilated in that keg. So that will give you complete this thing regarding the drug, regarding the how it works, its metabolic pathway, complete details regarding the protein molecule. You can get it from kegs, the 19 different databases which are available. And they are all, you don't need to worry about the reliability of the data, 100% reliable. Okay? So probably the first part is related to peptide therapeutics, and the second one is related to, second one was related
3D
to
brain.
Yeah, brain. That's what I said. Clarity, CLARITY.
Oh.
So where the three-dimensional image I showed you also, the neurons, the image which I showed you is actually the three-dimensional image of the neurons and the dendrimers or the dendrites of the neurons. So you can imagine how beautiful it will be if somebody is working on that. You can even use that concept to study, because Alzheimer's or schizophrenia, they're all related to what happens inside the brain, inside each neuron. That's what I said. Single cell study is so important. So microfluidics and all these things. Now today it's all gone where you study a population of cells. Because if you study an entire population, the data which you get may not be representative of what is happening in one cell. So a cancer cell may behave differently from other cells. I hope it's clear. So study has become more focused, single cell study. And there again, as I told you, if you're able to observe individual neurons and what is happening to the active, how the impulses are conducted. Based on this, you can study how a drug is acting on schizophrenic patient or Alzheimer's patient, how it is rectifying the neurons. All these things can be easily studied. Is it okay, madam?
Yes, thank you. Any other questions?
Can you give some light on space technology, which is going to change the way the pharmaceutical formulations are being made? Space technology, the pharmaceutical manufacturing, or the drug delivery system production, or vaccine production, whichever it is.
Okay.
Advancement in the space manufacturing.
Advancements in space. Again, I think probably we need to regard the use of pharmaceuticals, probably nanotechnology and related things you can extrapolate, because slowly they are trying to study how matter behaves in the space. Like, for example, I'll give you what is happening in the space. I can tell you some current information I can share, sir. That is on 3D printing. 3D printing on various organs. You can print any organ by this 3D printing concept. So this 3D printing concept is slowly being tried, and it has even been done. Almost 274 days, the particular cosmonaut or the astronaut was there in space. And during this 275 days, they have been able to print out so many organs using three-dimensional technology. So this is one thing. I don't know about manufacturing of pharmaceuticals exactly, but even these organs and medical devices today are also included in pharmaceutical category. Latest FDA categorizes even devices and stents, as well as even your organs, right? These are also going to be 3D printed organs are also going to come under the category of pharmaceuticals. So this was, in fact, for those people who are interested in studying more, you can study related to this thing in BBC website, BBC and CNN website. I have found out this related to this thing which I told you about studying in the space. So this 3D printing I told you about. 3D printing of several organs has been done in the space, because the main advantage of studying in space is the gravity is not there. So once the gravity part is not there, the cells do not crumble. When you do carry out the work in Earth, when you do the same study in Earth, the cells will crumble. So it becomes a hindrance to the study. But when the same study is carried out in space, then the cells are retained properly because there's no gravity there. So this is the advantage which they have quoted in that particular paper. 3D printing of several organs has already been done. It has already come into that, I think BBC interaction video, I think I saw that. It's a sufficiently latest area only. Thank you, sir.
And one more thing, there's a lot of companies are coming out with data collection. The data science is going to be one of the very important way of collecting all the data pertaining to your disease as well as some of the patients.
Yes, sir.
For drug development purpose. The way it's going to change the doctor's the treatment profile and how the patient is going to progress.
Yes, sir.
So many companies are coming for data collection, and how this data collection is going to be useful for them, as well as going to be useful for the medical fraternity.
Yes, sir. So here again, integration is most important. Like, for example, antibiotics surveillance programs. Every hospital, every district centers should come out with the interlinked thing of which organism is prevalent in that particular area. And this has to be connected to the state level centers. And that has to be connected to the central center, national centers. And those have to be centered through global centers. Only then, so there has to be a complete blueprint of what exactly is the thing happening at the minuscule level. Right? Only then you can really make meaning of what exactly is. Otherwise, you will get so much of data, but you will not be able to really interpret the data. I hope that's what I explained about multi-drug resistant organisms and tuberculosis therapy, DOTS therapy, and all those things. If you're able to link it to a computer, that is able to identify. Even I told you about how whether a patient is taking the medication or not, you can use an advanced capsule to find out whether the frequency of medication is being taken correctly or not. So there is a beep sound or something through a application generated this thing which the patient is able to come out with. He's aware of it and he takes the particular drug. So all technology driven. All technology driven.
Okay, thank you, Gibson.
Thank you, sir. Students, any more questions? Or anything which I can discuss at least?
There are no other questions.
I think no other questions, madam.
If there are no other questions, then we can conclude the session. Thank you very much, sir, Dr. Krishnan, sir. It was very lucid presentation and we really enjoyed from the university side.
Thank you, madam.
And all the interactions were so healthy and we were wondering, one thing, sir, how about the cost, sir, when it is used to the therapeutic purpose?
And that's why after so many years, it takes 20 years of research experience and you come out with one or two. It's all based on funding and based on commercial implications. And I told you about antibody and drug conjugates. So far, almost 10 years, research has been going on extensively throughout the world, but we have got only 2 FDA approved and even these smart capsules and all, only 1 or 2 have been really in market. They're all in the clinical.
In the market, how about the price if it is being used for the patient?
Unless the number of this thing is increased, only if you go in for number, you can reduce the cost.
Reduce the cost.
Yes, madam. That is self-explanatory.
I know.
Yes,
yes, sir. Thank you very much, sir. It was very nice listening to you. And thank you, Dr. Ravi, sir. Thank you, Dr. Sampath Kumar, sir. And thanks, Dr. Grace Rathina, madam, for moderating, both of you moderating the session. And I thank everyone who have joined through Zoom and through YouTube also. We had around 40 participants listening through YouTube. And thank you very much. And happy Diwali for everyone who's there in the session.
Thank you. Happy Diwali, all of
Thank you, madam. Thank you, madam. Thank you,
madam. Vice Chancellor, madam, also. Please.
Definitely, sir. I will convey to madam.
Thank you.
Thank you, man. Thank you, madam. Thank you all.